It's January the 21st, 2021, and welcome to the Water Action Platform. We finished last year on a high with our Christmas special, in which we showcased some of the excellent work the aid sector has done over the past year. We also had on LinkedIn our 12 Days of Christmas Advent Calendar, which has been viewed by literally thousands of people. Now, I'm not sure whether the attraction of the Advent Calendar was the 12 key learnings from last year's Water Action Platform webinars, or the fact that each day ended with my colleague, Yang Villa, on his ukulele singing a gloriously uplifting Christmas tune. I guess it doesn't really matter. But thank you to all of you who made donations to our charity appeals for WaterAid and Happy Tap. Isle has matched those donations, and your contributions will help make things better for those less fortunate during 2021. I also thank all our sponsors for their ongoing support. 2020 was a dark year, but we finished it with hope of new vaccines. And today, we're going to delve a little deeper into exactly what that means. Dr. Joe Burgess will share what it's like to be a guinea pig in a vaccine trial and what some of the variants are which we need to know about. If you're watching this at 7:30 a.m. UK time, then this is a live broadcast and there will be Q&A at the end. So if you want to raise a question, please just post it in the question box by clicking on the triangle to the right of your screen. Now, following feedback from you, the focus of our sector expert and technology showcases today is on emerging contaminants, in particular PFAS. But before we get to that, let's start, as always, with a quick look at some of the water sector related stories around COVID-19. Now, to my rather simplistic mind, I see the following as one piece of good news and one of bad. So first, the good news. In something which you arguably might have expected to see on an episode of the 1980s classic The A-Team, we have the development of low-cost passive samplers made from everyday household items. Now, we all know that the monitoring of sewage for wastewater-based epidemiology could help in the fight against the virus, but one of the challenges has been how to gather samples. Traditional sampling methods like auto-samplers or grab sampling are not always appropriate, in particular for small rural works. So, Schlang et al. Have both developed and validated a fantastic array of cheap and easily deployable passive sampler units. Now, just look at these photos. They are made from readily available consumers. I just love the colander style one. That is my favourite. I can hear now the loving and supportive response from my wife, were I to suggest that I use kitchen equipment to gather sewage samples. Now, joking apart, this study provides the first evidence that simple, cheap, passive samplers can be used to detect SARS-CoV-2 in wastewater. The bad news story, however, is that in certain parts of the world, we are now seeing high levels of SARS-CoV-2 in river waters. Now, this is happening in countries where large quantities of raw sewage are being discharged into rivers. The example presented here is from Ecuador, but they're by no means an exception. Many countries have similar treatment challenges. So in Quito, the capital of Ecuador, less than 3% of the sewage is treated before discharge. Now, this means that the level of SARS-CoV-2 found in the urban rivers around Quito was similar to that which was seen in the raw sewage in Valencia, Spain, when it had 5,000 active cases, and similar to the levels in Paris when it had 10,000 hospitalized cases. Now, intriguingly, at the time of sampling, there were only 750 recorded COVID cases in Quito, which suggests a large number of undiagnosed cases. This further highlights the importance of rolling out universal sewage treatment programs, which will serve all communities across the world. Now, as mentioned earlier, in this next section, Dr. Joe Burgess will share some insights into the vaccines and their variants. She will also share what it's like to have been a vaccine guinea pig. Now, as the vaccine news is literally changing almost by the hour and certainly by the day, we are doing today's broadcast live so we can give you the absolute up-to-date latest news. Jo is currently based in South Africa, working from her home. If you have any questions for Jo, raise them in the chat box to the right of your screen and we'll address them at the end of the webinar. Jo, I'm hoping the internet is working. Over to you.
I'm hoping so too. Thank you, Piers. It's great to be back. Especially with the added excitement of a live webinar over an African internet connection. So I thought I'd spend the next few minutes to try and answer some of the key questions people have asked me over recent weeks. Firstly, 9 vaccines have been approved so far, and this is good news, and the big 5 are these on the screen. You can see they use 3 different biotechnologies to achieve immunity. The technologies are all very remarkable. They're not all completely new, and they've been tested so far for adult and geriatric application, but they haven't so far been tested for use on children. The developmental and reproductive toxicology studies are underway, the DART studies, and these are conducted to assess the potential risks associated with the vaccines to a developing fetus, so we can work out whether we can vaccinate those who are pregnant. Uniquely, Johnson & Johnson, or Janssen on this table, are undertaking 2 phase 3 trials in parallel, and they're finding out whether they can use a single injection to raise immunity instead of 2 injections. And this is a complete game changer because it less than halves the logistical challenges of a vaccine rollout programme. To obtain approval, even for emergency use, the developers of these vaccines have to publish their data sets for peer review. Usually that happens in one of the big medical journals like the Lancet or the New England Journal of Medicine, and they also have to go before the boards of the regulatory agencies for a data defence. The minutes of the US FDA, Food and Drug Administration, and European Medicines Agency meetings are always published, so anyone who says the results are hidden or they don't know what's in the vaccine just hasn't looked in the right places. This table carries links to all of the data. But be warned, Pfizer's report is 92 pages long, and the FDA report on the same drug is 53 pages long, and yes, I read it all. This is what the data look like. On the y-axis, you have the total number of participants in a phase 3 trial, and this trial has 60,000 people enrolled in it. The y-axis tells you how many of them have caught COVID-19 since the trial began, so since they received an injection. The blue squares show you the number of people who were unfortunate enough to receive a placebo, so a sterile saline injection, and the red circles are the people who were given the vaccine candidate. The inset to the left shows you just the first 3 weeks in a smaller graph, and this is a good indication of the number of participants who were infected but were pre-symptomatic when they went for their first injection, so on day 0 of the trial. And it takes about 10 days for the control group, the placebo group, and the vaccine group to diverge. So what about the new variants? Obviously, we're racing against time with the vaccine, against the number of infections, and the news outlets are talking a lot about what they're calling the new variant. Now, I don't want to alarm anyone, but this phylogenetic tree shows about 3,000 of 12,000 or so variants of SARS-CoV-2 that have emerged since the original strain was detected in China a year ago, and this is completely normal. So all of these dots across the timeline on this chart show you a new variant, a new strain emerging and being identified. SARS-CoV-2 mutates randomly, and it happens once or twice a month, and most of these mutations are completely inconsequential. Some of them give the new variant an advantage, like increased transmissivity, and there are plenty of other viruses like polio that also mutate at this rate, and we successfully vaccinate against them. All of the vaccines are designed to cater for mutations. So of those 12,000 variants, only 3 have been identified so far to have the potential to make it necessary to tweak the vaccine design. And if a mutation does render the vaccine or more than one vaccine useless, the vaccines are incredibly easy to change. It's like editing a Word document. You just change a few words, you tweak the code a little bit. You don't have to rewrite the document. So the vaccines instruct the body to generate numerous different antibodies that act against the virus. We have a range of nucleic acid vaccines. These include the mRNA vaccines, the Pfizer and Moderna vaccines, and the other ones on the table are DNA vaccines. Just yesterday, news was announced of a South African variant called 501Y.V2 variant, which evades 3 classes of monoclonal antibodies. But the vaccines induce a polyclonal response, so numerous antibodies that connect to different parts of the virus. So if a tweak is needed, the regulators don't need phase 1, 2 and 3 trials of the same vaccine again, because the Word document, the code, isn't changed enough to affect the safety of the vaccine. Now, I'm going to spend a few minutes just talking about what's happening in the Johnson & Johnson trial, where I'm a volunteer for the phase 3 single-shot trial. There are 45,000 volunteers in this trial in 9 countries across Europe, Latin America, Asia, and Africa. And to begin with, it starts with an awful lot of paperwork. The consent form is 27 pages long, and they go through it with you in very fine detail to make sure you know what you're getting yourselves into. Then you undergo a range of health tests, things like blood pressure, weight and height, which I thought was a bit cruel at Christmas. If you're female, you have a pregnancy test, and if you're male, a prostate functionality test. They measure things like cholesterol and HIV status, and they also do an antibody test to make sure you don't have COVID-19 or you haven't had it at the time of the injection. Assuming all the test results come back the way they want, you receive an injection in your upper arm, and it's either saline or the vaccine, and it's a double blind, so you don't know what you're receiving and the researchers don't either. And then you get given a goodie bag, so I've got my very own pulse oximeter and thermometer, and I had to measure any side effects from the injection for 8 days after the jab took place, and I report any side effects on an app. Then twice a week for the next year, I have to log into the app and tell them whether or not I've caught COVID-19. And for a second year, so a total of 2 years after the study, I have to do that twice a month. And basically that's it. You get an injection, you go about your life as normal, and you tell them if you become ill. The data are reported when a threshold number of cases are reached, and the study stops in terms of safety data after 2 years. So when 170 people in the group in total have caught COVID-19, and we hope they're all in the placebo group, then Johnson & Johnson can report the results. And after 2 years, they can make a conclusion on whether or not there are any undesired side effects. So this tells us we're in the beginning of the vaccine endgame now. We're into regulatory approval and actual distribution or rollout of vaccine programs into millions and millions of people. And we need people to take the vaccine faster than they can catch COVID-19. Vaccine hesitancy is real and it doesn't help. We're going to see the usual run of mortality and morbidity that you would see across large samples anyway. If you took 10 million people and just waved your hand over their upper arm, in the next 2 months, 4,000 of them would have heart attacks, 4,000 of them would have strokes, you'd have 9,000 new cancer diagnoses, 14,000 of them will die anyway from causes completely unrelated to whether or not they had a vaccine. We're literally going to have cases where someone gets a vaccine and drops dead the next day, and they would have done anyway. It's going to be really hard to convince their friends and family, though, that it's not related to the new drugs. On that note, I'm handing back to Piers in London.
Oh, thank you, Jo, for that excellent from the coalface insight. There will be Q&A session with Jo shortly. However, I now have a few short updates. Firstly, last month, Aegea in Brazil hosted a virtual tour of their digital twin. Now, the full recording's on our website, and you can see here a flavour of what the tour involved. Now, I've been inundated with comments from people saying how impressive and useful the tour was, so thank you, Aegea. And I believe we may have some future tours of other digital twins planned, so watch this space. Next, there is a heads up of news of a World Bank organised webinar on resilience during COVID-19. Now, this will be the second in a series of 3 webinars. The first was for World Bank clients only. The second will be in April and has the title COVID-19 and How to Beat It, Technologies to Detect, Destroy, Monitor and Manage SARS-CoV-2 in the Water Industry. We will, of course, report back on it here, but this event is open to all, and once the date's finalised, we will share it with you. Next, we have the Diversity and Inclusion Forum. As you can see here, we are a sector which is, in general, very poor at diversity, be it on gender, colour, creed, age. Last September, about 40 organisations from across the Water Action Platform membership got together to share data and learning on how to improve diversity in the water sector. The invitation here now is for you or your colleagues to join a 6-monthly update session to be held next month on this important issue. I will share the relevant links on my email when I circulate this slide deck after the broadcast. Now, my final update relates to the £200 million OFWAT Innovation Fund. Now, OFWAT is the financial regulator in England and Wales, and they've basically created these 2 competitions, the Innovation in Water Challenge, which opened on the 18th of January, and a second larger competition, the main challenge, which opens in the spring. Now, the Innovation in Water competition provides an opportunity to access between £50,000 and £250,000. Technology companies can enter the competition, but it has to be done in partnership with one of the 17 OFWAT regulated water companies. Now, for more information, visit the website listed here. Rather helpfully, it also provides the relevant contact details for the water companies with whom you might want to connect. So we now come to our sector expert section, and this week it's Carol Walcyzk, Vice President for Water Quality and Compliance at Suez North America. Now, our focus this week is on emerging contaminants, and in particular polyfluoroalkyl compounds, often referred to as PFAS. PFAS chemicals are man-made compounds found in many consumer products, in particular fabrics, cleaning products, paints, and firefighting foams. Their ability to remain intact for long periods of time mean that they build up significantly in the environment, and there are growing concerns about the health and bioaccumulation effects. As a water sector, we need to be aware of this issue and proactively respond to address it. Now, North America has arguably tended to lead the thinking within the water sector around how we manage these emerging contaminants. So last week, Carol spoke with the president of ISLE Inc., Christina Armadpour. Christina opened by asking Carol which of the CECs, the constituents of emerging concern, were of most interest.
So right now, PFAS is a pretty big problem because for us as water utilities, because the public concern and the regulations are advancing so much faster than the technology and the funding and the science. So we have regulations being driven by public outcry and not by science and not by technology. So what's happening is we have different states setting their own limits. They don't want to wait for the federal regulation, and they're not following that federal, in general, they're not following that federal process of cost benefit analysis, feasibility, treatability, and looking at all the occurrence data. They're looking at the occurrence data for a very limited subset and regulating a larger set. So that's a big challenge we're facing. Our regulated utility operates in 6 states, and all 6 states have different limits, and including neighboring states, which can be very difficult to explain to customers.
How are you striking that balance between what is regulated, where you're heading, where you think the industry is heading, versus and all the other challenges that your organization as a water utility is needing to prioritize? How are you striking that balance?
It's not easy. We're trying to be proactive in a way that is not a way the water industry is used to being. We're used to having 6 or 8 or 12 years to prepare for a new major regulation, and when we started seeing that we weren't going to be given the courtesy of that kind of time frame, we started testing everywhere, even where there was no pending regulation, just to see. Many of our facilities were not regulated by the unregulated contaminant monitoring rule. We had no baseline data, or even if we did have unregulated contaminant monitoring data, it was at that 20 part per trillion, 40 part per trillion limit. So we just developed, kind of a snapshot of where we are so we can identify risk. And same thing with wastewater. There's no pending regulations, but we just took a look with the drinking water methodology to see if we could have potential risk. And it was not an easy sell. These samples cost about $500 each when you count the trip length. So it was challenging, but it's important information to have.
What are other parts of the globe that you yourself, being an expert in this field, where are you looking to learn from your peers and what's happening in other parts of the world with other CECs?
It's interesting. In other parts of the world, they're not really, they're CECs, but they're not, it's more the emerging concern part because they're not new. There's a lot of concern about manganese and aluminum and contaminants that we consider aesthetic, because there have been potential health effects linked to them with Alzheimer's and some cognitive issues with manganese. So they're keeping a very close eye on manganese and aluminum and actually setting internal limits within at least within Suez. We have internal limits for those, which is not, it doesn't seem to be on the radar at all here in the US. I know manganese is in the current unregulated contaminant monitoring rule, and they may be considering making it a primary contaminant, but it's got a much higher profile everywhere else in the world, which I thought was interesting. But other things like PFAS definitely is not considered a high priority in most of the rest of my company's service area. And things like disinfection byproducts, it can be all over the place. What's allowable in Morocco is crazy compared to what might be allowed in France or here.
Well, thank you, Christina. Thank you, Carol. Now, the full interview is available on the Water Action Platform website. As Carol noted, we're in uncharted territory, trying to find contaminants which may not yet have regulations controlling them. Yet we need to do this work in order to build a reliable and robust picture of the risks so that we can respond appropriately. Now, this isn't easy, and it's not cheap, and it's not something individual utilities can do alone. We must share our learning so that everyone across the sector can benefit. And this is precisely why we're raising it here today. Now, ISLE has recently completed a project identifying the early stage and emerging solutions that not only look at the treatment and removal, but also the destruction of PFAS. If you'd like to know more or would like to be connected to Carol or Christina directly, well, as always, let me know. So we now come to our technology showcase section, and staying with today's theme of emerging pollutants, we have 3 technologies which specialize in addressing that issue. We start with Cerafiltec from Germany. Now, Cerafiltec is arguably the most innovative ceramic flat sheet membrane module in the world right now. The key thing here is that ceramic membranes have so much more to offer than conventional UF membranes. For example, they allow for the combination of absorption and filtration in one step, enabling the selective removal of dissolved contaminants such as PFAS. Now, as explained in this short video from the COO Julius Gloeckner, which was rather impressively filmed last week in their offices during lockdown on a mobile phone, Cerafiltec has demonstrated that they can remove PFAS to below detectable levels from high initial concentrations.
Our ultrafiltration is based on ceramic flat sheet membrane technology. It is not just ultrafiltration. We combine filtration with adsorption, and this allows us to remove even dissolved contaminations such as radioactive isotopes, molecules, odor, taste, color, dissolved organics, and also PFAS below detectable limits. The membrane is an out-to-in filtration membrane. That means it's fully submerged in water. The water penetrates both sides from the membrane out to in, gets collected in these water channels, and gets sucked in operation out of this nozzle. One module then has 34 of these full membranes that have end caps and nozzles on both sides. The water flows through the end caps, gets collected here in the front side of the module to the side, and flows out to the top to the main header pipe. And the backwash works exactly the other way. The water gets pushed into the module and into the membranes. But how do we remove PFAS with an ultrafiltration? Well, about a decade ago, we invented a process by which we are layering the membrane with a very thin layer of an adsorbent to selectively remove dissolved contaminations. For example, for removing radioactive isotopes, we're using a manganese-based layer. For removing PFAS, we're using powder activated carbon as a layer on the membrane. During operation then, this layer gets replenished after every backwash. This is a membrane coated with powder activated carbon. Evenly distributed on the membrane surface, very thin, as you can see. And during the filtration process, every water molecule needs to pass the powder activated carbon layer before it enters the inner membrane channels. During this process, every PFAS molecule gets absorbed by the powder activated carbon. And once the powder activated carbon layer is fully saturated and cannot absorb any more PFAS molecules, we simply perform a standard backwash to replenish the layer and start filtration again. And how easy it is to remove the layer, you can see here. It is not sticky. So why is this special? Well, finally, it all comes down to cost. And this is where ceramic flat sheet membrane combined with powder activated carbon yields much better economics than other solutions, such as GAC, ion exchange, or reverse osmosis. Everyone knows the smaller the particle size and the closer they are together, the better the adsorption effectiveness and efficiency. And we simply need 3 times less of the adsorbent. Further, we are achieving a recovery rate of up to 99%. This means a very small waste stream, hence low disposable costs. And this process has been proven. 600,000 cubic meters of daily plant capacity have been installed using this process. And this is why we firmly believe that ceramic flat sheet membranes with powder activated carbon will become the standard in removing PFAS.
Excellent. I love that gazing into the distance as he asked his own questions. Now, Our second technology is from an Australian company called Infinite Water. They've developed the Hydroxon process, which is a catalytic advanced oxidation process to remove metals, dissolved organics, and other refractory, toxic, and non-biodegradable compounds. Hydroxon is a low-energy solution. The manufacturers claim that the total energy consumption of a hydroxon unit is less than 0.1 kilowatt hours per meter cubed, and it can replace several processes in a conventional treatment train. The technology has an initial conditioning stage prior to a catalytic oxidation reactor. So here is Joshua Pinto, Sales Director, explaining how the process works.
Hydroxon essentially is a two-step process. The two steps are conditioning and catalytic advanced oxidation. Conditioning might involve settling of suspended and colloidal solids through normal processes like flocculation and coagulation. And we like to use a metal salt like ferric chloride, for instance, as the coagulant. And the reason is, one, it provides a very good flock, but two, the residual ion could be used as our catalytic metal when it hits the reactor. The second thing in conditioning stage is a bit of pH adjustment if required, and the adjustment of ORP, because we need a very high oxidative environment inside the reactors. Our catalytic advanced oxidation reactor vessels is actually a mixed media filter. So it does produce the turbidity requirements that you need as your critical control point. So we use special catalytic media as the filter media in our vessels. So now when the conditioned water comes into contact with the catalytic media, the residual metal, which is iron in this case, or could be manganese or aluminum, transitions from a lower oxidation state to a higher oxidation state, and in the process releases the hydroxyl radical. The metal then goes back into the solution and comes back into the contact with the catalyst, and as it moves down, the reactor vessel produces an abundance of hydroxyl radicals. So as a result, pathogens are removed, metals are precipitated, and organics are degraded. The Hydroxon process uses only 100 kilowatt hours per megaliter. Now that is the energy used for direct media filtration. So you now have the benefits of direct filtration like low energy, small footprint, but the power of advanced oxidation. The processes of sedimentation and flocculation, ozonation maybe for organics removal or color removal, media filtration and adsorption for specific metal removal, softening or even membrane filtration like ultrafiltration, and then reconditioning could all be replaced by the Hydroxon process. So one, that means a shorter treatment train, lower pumping costs. But I think more significantly, what this says is that the Hydroxon is a parallel process, and it's able to decontaminate the water from those contaminants that are not being measured or not known to exist in the water matrix.
Our last technology is Pharem filtration systems from Sweden. Now, this is also a low-energy enzymatic filtration system specifically designed for removing organic micropollutants. Their systems gravity-fed and can be fully functional with little or no electricity input because the enzymes don't require additional energy to do their thing. We have, as usual, a short video prepared, but unfortunately we've not been able to connect with Pharem to let them know that we were going to talk about them today. So as soon as we hear back from them, we will post it on our website. However, as always, if you want to know more about any of these technologies, Hydroxon, Pharem or Cerafiltec, just let me know. Now, our end finally this week is the live interview with Joe Burgess. If the technology allows us, we should be able to go to a double screen. Joe, are you there?
I'm here and I hear you loud and clear.
Oh, that's brilliant. I am so pleased. Now, we've also got in the background, we've got Megan, who is monitoring the questions, and I've now got them here on my phone. She's screening them. So if your question doesn't get answered, you can blame Megan, not me. So, Joe, I'd like to start. We've got a question from Caroline. Are there any safety concerns over the speed at which the vaccines have been developed?
No, the short answer is no. The process that the vaccines have gone through, so the phase 1, 2 and 3 trials, is exactly the same as they always have done. What's made it possible to do everything so much more quickly is, first off, there's been regulatory buy-in because of the number of people dying. So the approval, the red tape that normally takes months to wade through to get permission to undertake and recruit volunteers for phase 3, for example, was forthcoming really quickly. And the second thing is investment. Normally it takes years to raise enough investment for a vaccine, particularly if the market is quite small.
So governments have been throwing money at it this time round, haven't they?
Governments and the private sector, the chances of not having a big target market, 0.
Excellent. All right, well, I've got a technical question here from Andrew Walker. Could you give us a comparison between DNA and mRNA?
Sure, okay. The Pfizer and Moderna vaccines are mRNA, and that means messenger RNA vaccines. And the AstraZeneca and the Johnson & Johnson and the Sputnik vaccines are DNA. So they're all nucleic acid vaccines. And they all work like messages carrying a picture into your cells. And that picture shows them what the spike protein or a range of spike proteins, the same as the one SARS-CoV-2 looks like, and instructions on how to kill it. Now, mRNA is designed, it's really fragile, and it's designed to disappear really quickly. So like a Snapchat message, you look at the picture, then it's gone. But your memory T cells in your immune system remember what that picture looked like, and they remember the instructions, and they carry on making the antibodies. The mRNA vaccines are encapsulated in a lipid micelle, a fat bubble, and that's what's been causing the occasional allergic response to those particular vaccines. The DNA vaccines use DNA instead of mRNA, and they take it from an adenovirus. So it's a completely harmless virus that gives chimpanzees common colds, but doesn't infect humans. And they use DNA to carry the instructions into the cell. And your cell then makes the spike protein or spike proteins itself, the same as the ones on SARS-CoV-2, and your immune system raises antibodies against it. DNA is much more robust as a molecule than mRNA. It's not designed to break down the way mRNA is. And that's why the DNA vaccines don't need to be held at minus 70 degrees for storage and transport, and the RNA vaccines do.
All right, so actually, there's a second part to this question, which is which one would you recommend to your grandmother? And you would just, well, which one would you recommend to your grandmother?
It depends what Granny's allergic history is.
I thought you were going to say it depends whether you like your granny or not.
No, not at all. So far, the results that have come in have all been between 90 and 97% effective. So all the vaccines that have been approved are about as good as each other in terms of efficiency. They're all very solid biotechnologies, although mRNA looks new. It's a vaccine delivery system that's been in development and under research for 20 years or so. They're all safe. I would let Granny have whichever one she could get the sooner she could get it.
So it's about access rather than being too selective. Okay, so Monya from South Africa has given 2 questions. I'll read them both to you so that you can decide how you want to answer them. Can you perhaps explain why the need for ultra cold storage, minus 70 degrees C, for some of the vaccines? You sort of just touched on it there. And will COVID-19 vaccination become an annual requirement?
Good questions, both, yeah. The second one is possibly. It's difficult to know yet because we don't yet have a good handle on how many of the mutations are going to cause the vaccine design to need to be changed. So the reason every winter there's a new flu vaccine is because every winter there's a new strain of flu that's evading the vaccines which were previously on the market. And it may be that COVID-19 turns out to be the same as that. Currently, there are only 3
It just becomes endemic
strains.
and something that we just have to manage.
Yeah, exactly. Yeah. It becomes endemic. It may or may not become seasonal. We don't know yet. But the reason the mRNA vaccines need to be held at minus 70 is because the mRNA itself is so fragile. And that's why the Johnson & Johnson one in particular is a complete game changer. If it is a single shot, then the logistics of putting it in the back of a van, it doesn't need to be at minus 70. You can take it out to a rural village and give it to a really vulnerable population. And you don't have to go back. If you visit the village, you vaccinate everyone, you're done. That would be so much easier than the ones that require 2 doses and cold storage.
Yeah, well, Tim Jones has raised a question around, well, it might be easy to change the vaccine, but is it easy to re-administer it? And I think you're touching on that point there. Let's keep it here. So I think, how are we doing on time? I think we'll make this probably the last question. So a question here from Karen. We're fortunate in the UK that we have many doses of the vaccines and it's being rolled out. How can the inequality in the availability of the vaccine in the world. I read that some low-income countries only have a handful of doses. Do you have a view on how this inequality could be addressed?
Yeah, the COVAX program, which the UK invests in to a very large extent, and other countries, is trying to address some of that inequality. But there's been a lot of chat about the ethics of less vulnerable people in rich countries being vaccinated before more vulnerable people in low-income countries. Part of that is going to be accelerating the rollout of the DNA vaccines, like the Oxford AstraZeneca one, like the Sputnik, like the Johnson & Johnson, where we can reach people in low-income countries, because the other attribute that DNA vaccines have is they are a fraction of the price of the mRNA vaccines. So we're looking at £25 a throw for mRNA vaccines and about £3 for DNA. And particularly if you need 2 doses of the £25 one compared to 1 dose of the £3 one, it suddenly becomes much easier to make it affordable for people in vulnerable low-income communities.
Brilliant. We're up against time, but Joe, brilliant to have had you join us. Just before we hang out, is there anything else you'd like to add?
If anyone has questions that they didn't get to ask, if you're in the WhatsApp knowledge hubs, then by all means contact me that way, or by email if you prefer.
Thank you. Well, thank you everyone for your questions, and Joe for your thoughtful answers. Now, if you enjoyed the live Q&A element, please let me know and we can try and repeat it in the future. It adds a certain technical excitement on our end here, as you can imagine, and I can't believe that the South African internet didn't let us down. We've got to repeat it again in 8 hours' time, and I think the storms in South Africa in the afternoon are going to add an interesting little dynamic to it. Now, on next month's webinar, the focus for the sector expert and technology showcases will be on biosolids. Now, perhaps I'm biased because my background is in sludge treatment, but I am particularly looking forward to it. Thank you again to all our sponsors. The next webinar will be on Thursday, the 18th of February. You'll receive a calendar invite shortly from Megan. In the meantime, keep asking questions, keep sharing, and keep safe.